Hello viewers, good morning. The topic that we are going to look at today that is mushroom diseases. When mushrooms are growing in the cultivation site, it can be affected by many microbial diseases. The diseases of mushrooms are mainly caused by bacteria, virus and fungi. In this video, you can see in the detail about the disease spread by bacteria. The next bacterial disease on mushroom, drippy gill. Drippy gill disease was first reported in the year 1968. It is a serious outbreak of infection in all edible mushroom during cultivation period. How can we identify the drippy gill disease? The small drops of droplet that present in between the bridge edge of the gills. The drops full of bacterial pathogen that is comes out from the gills part. The bacterial droplets developing on the gills, that is the characteristics of the symptoms of drippy gill. Hence it is called the drippy gill. And the drippy gill disease is caused by Pseudomonas agarici bacteria. The discrete bacterial droplets on the hymeneal lamellay and coalesced to form a ribbon of bacterial ooze. The Pseudomonas agarici bacteria that colonize the extrahyphal spaces and degrade the extracellular matrix and reduce the strength of the fruit body of mushroom and make splitting of hymenium. The Pseudomonas agarici can produce some toxin against Agaricus bisporus species and enter into the protective membrane and infect all the parts of mushroom. The gills and lamellae of matured mushroom can develop up to 2 mm or more. At the center of the gills, it consists cream or cream gray droplets may predominantly on the gill surface. The droplet size may increase up to 0.5 to 1 mm in diameter. So it can connect these spaces of in between the gills. The droplet overfilled with the bacteria. When we see under the UV light the bacterial slime or the droplet, it will appear like fluorescent blue in color. It inhibits the graft development and deterioration of the pileus and destroy all the total grafts of mushroom. And this is the morphology of Pseudomonas agarici bacteria. It is a gram-negative bacteria. It has very thin cell wall and highly present in the soil, hence it is called soil bacteria. It is a predominant bacteria in soil. And the rod-shaped bacteria, slender bacteria, and the wide 0.5 micrometer to 0.8 micrometer in diameter and length 1.5 micrometer to 3.0 micrometer in diameter, and motility that moving by the polar flagella. Sometimes it has more than 2 flagella may be present. And these are the examples of bacterial diseases on mushroom. The blotches disease were classified by the different colors on the pileus of mushroom. Examples: brown blotch disease, ginger blotch disease, and other blotches. Brown blotch disease. The color can be characterized by the brown color, chocolate brown color appearance on the pileus of mushroom. And ginger blotch disease also appear as ginger color and other blotches, that is discolorized spots that can appear on the pileus of mushroom. And brown blotch disease caused by Pseudomonas jolasi, ginger blotch caused by Pseudomonas gingeri, and other blotch caused by Pseudomonas fluorescens, Pseudomonas aeruginosa, and Constantini. And this is the drippy gill, that means the bacterial pathogen that can ooze out from the gills part of mushrooms. And the drippy gills majorly affect the species of mushroom, that is Agaricus species and Pleurota species, especially from Agaricus species, Agaricus bisporus and Agaricus campestris, that is button mushroom. And the next species, Pleurotus astratus, that is Asher mushroom. And these are the symptoms of drippy gill. Cream grey colored bacterial droplets are observed on the gills of mushroom in the early stage. These droplets are surrounded by the dark brown to black spots which increases in their size so up to 2 mm or more. These droplets may present the gap between the gills part of mushrooms and the total strip color becoming like brown in color. When we dissecting, the young button mushroom may reveal the droplets and gills prior to opening, and the fruit body of infected mushroom it become abnormal shape with different lesions may appear on the total part of mushroom. The lesion color will be like black or brown in color, and the total surface lesions that can be discolorized. The whole plant it become produce some bad odors or unpleasant odor. How to control and prevent drippy gills disease during mushroom cultivation? First, drippy gill caused by Pseudomonas agarici bacteria. These Pseudomonas agarici bacteria was majorly present in our environment, especially in soil region. This is the predominant bacteria in soil. But mushroom cultivation we need some basic requirements like paddy straw, compost and calcium soil that is collected and prepared from outdoor only. So there is chances the bacteria already presented in the compost, calcium and paddy straw. So we give much more attention to compost and calcium soil preparation, and we should use proper pasteurized compost calcium soil and use proper sterilized paddy straw and use sterilized equipments. While the watering irrigation we use chlorinated water, which the chlorinated water consists calcium hypochlorite and sodium hypochlorite. When we follow this instruction, so we can strongly destroy the drippy gills during the mushroom cultivation. Thank you.